Okay, so let's continue with the pathogenesis or mechanisms of neoplasia. Basically, we are going to talk about oncogenesis, and this is the molecular basis of cancer, right? So, oncogenesis, remember the basic principles or the general principles. Non-lethal genetic damage is the most important initiating event. Yeah, so mutations may be inherited in the germline or acquired somatically, whether chemical radiation or microbes, and tumors form by clonal expansion of a particular gene or single genetically damaged cell could lead to cancer. And of course, genetic damage involves four main classes of regulatory genes, and we'll talk about that later on. So the pathogenesis of tumor, of course, genes are involved, as I said, and the growth promoting genes, which we already know as the proto-oncogenes, can be converted to tumor promoting genes, and the growth inhibiting genes, which are the tumor suppressor genes. Basically, they encode the transcriptional factors involved in regulation of growth. So, the four main classes of regulatory genes are what I'm talking about right now, and with the first being the proto oncogenes, and then the tumor suppressor genes, and also they are also regulate genes that regulate apoptosis, and also genes that repair DNA damage. Those are the four main. There are several others though, but these are the four main ones. Yeah, so with the growth inhibiting genes or the tumor suppressor genes, they encode transcriptional factors involved in regulating or regulation of growth, and there are genes that regulate apoptosis too. And there are also genes involved in repair of DNA damage. So loss of function of these um, allows progressive accumulation of mutations. So carcinogenesis is a multi-step process at genetic and phenotypic levels. So cancers acquire several other additional mutations, ultimately from point of mutation, and phenotypic attributes, for example, excessive growth, local invasiveness, distant metastases are acquired in a stepwise fashion called tumor progression. So the phenotypic attributes are gained by progressive accumulation of mutation and favored by defects in DNA repair. And the malignant phenotype, for example, sufficient, self-sufficient in growth signals, and remember, insensitivity to growth inhibitory signals too could um, could be a one of the malignant phenotypes, and invasion of apoptosis, defects in DNA repair, limitless replicative potential, sustained angiogenesis, ability to invade and metastasize, and also escape detection and destruction by immune cells. Yeah, so these are characteristics of um, or there are major malignant phenotypes that you can think of. And mutations in the genome of somatic cells could either lead to activation of growth promoting oncogenes, or alteration in the genes that regulate apoptosis, or inactivation of cancer suppressor genes or tumor suppressor genes. So what could lead to the mutations? Either inherited mutations, of course, genetic factors, and acquired or environmental factors, whether chemical, radiation, or viruses. So this could lead to the third pathway I talked about: activation of the growth promoting oncogenes, alteration of genes that regulate apoptosis, and inactivation of cancer suppressor genes. And all these could lead to expression of altered gene products and loss of regulatory gene products. And with this, it could lead to malignant neoplasm, but through which other submethods could lead to clonal expansion, additional mutations or progression, and then heterogeneity, and finally to malignant neoplasms. Alright, so with proto-oncogenes, they are normal genes that promote growth and they encode protein products that play various roles in the growth sequence, and also genetic damage converts them to more growth promoting genes called oncogenes. And the normal sequence of growth signal is that the growth factor binds to growth factor receptor, and a transient activation of growth factor receptor happens. There's a signal transaction or transducing proteins in the inner layer of the cell membrane, which sends message through cytosolic messengers across the cytosol to nucleus to induce and activate nuclear regulators of DNA transcription. And there's an entry of cell into and also propagation of the cell in mitotic cycle, leading to proliferation. Alright, so the growth factors binds to growth factor receptors, or signal transducing factors, and then finally DNA binding proteins concerned with transcription, and then cell cycle proteins. Alright, so with the growth factors, we have this SIS, INT2, IGF1, and the growth factor receptors, there are several: EP2, FMS, MET, TK receptor, RET receptor, EP1 receptors. And with the signal transducing factors, we have the G proteins for the RAS, GTPase activators, we have the GAPs, and then KREV, the membrane associated cytoplasmic kinases, SRCs, FGRYES, and then non-membrane associated cytoplasmic kinases like RAF, MOS, PIM1, FP. They're all signal transducing factors. And then finally, the DNA protein, DNA binding proteins concerned with transcription, because we know them to be heterodimeric, whether Fosjan or MYC, Max, MEX, or non-heterodimeric like MYB, REL, ETS, and also with the cell cycle proteins, the cyclin, for example, D1. So gene products of these are the major gene products of proto-oncogenes as steps involved in cell growth factors, growth factor receptors, binds to the growth factor signal transducing factors are being activated, DNA binding proteins concerned with transcription, and with the cell cycles protein and cell cycle continues. Alright, so with the protein products of oncogenesis, the oncogenes encode proteins called oncoproteins resemble the products of proto-oncogenes, but differ in that the oncoproteins are devoid of regulatory elements, and their production in transform cells is not controlled by growth factors or other external signals. So the oncogene produces a defective member of the pathway called an oncoprotein, which acts abnormally as a differentiation or control protein and leads to increased proliferation. And an oncoprotein may therefore be a growth factor receptor protein involved in signal transduction, nuclear regulatory protein, or the cell cycle regulator, like I talked about. So activation of cellular oncogenes, mutation of proto-oncogenes may lead to changes in the structure of the gene, resulting in synthesis of constitutively activated abnormal proteins, which is the oncoprotein, and changes in the regulation of gene expression results in enhanced or inappropriate production of structurally normal gene products. So point mutations in the proto-oncogene leads to an abnormal functioning protein, and these are some of the mechanisms of activation. That's in point mutation, whether substitution, deletion, or insertion. Right, and another thing too is chromosome rearrangement, and with that there are two types, whether translocations, which is very common, and inversions. Translocations activates the proto-oncogenes in two ways: whether over expression of the proto-oncogene by translocation, placing the proto-oncogene under the influence of regulatory genes for immunoglobulins or T-cell receptor loci, or translocation may attach normally unrelated sequences from two different chromosomes to recombine and form hybrid genes that encode a growth promoting chimeric oncoprotein. For example, in hematopoietic tumor, like chronic myeloid leukemia, Philadelphia chromosome has the oncogene formed by fusion of separate genes like CABL from CH9 and BCR from CH22. So when 922 translocation occurs, CABL BCR is formed that encodes a chimeric protein with tyrosine kinase activity. Right, so an example is translocation with proto-oncogene coming under the influence of a powerful transcriptional promoter. So chromosome, let's say 814, and then it's being moved from 8 to 14, and the that mechanism of translocation, mechanism of activation through translocation could lead to, let's say, conditions like Beckett's lymphoma. So inversion also refers to rearrangement that involves two breaks within a single chromosome with reincorporation of the inverted segment, and it may involve only one arm of the chromosome, which is paracentric, or the opposite sides of the centromere, which is pericentric. One arm is just paracentric, opposite sides of the centromere is pericentric, and the effects is similar to translocations. So gene amplification leads to an increase in product and can occur either intra chromosomally or extra chromosomally in the form of double minutes, and then so that's another mechanism of activation, whether amplification or trinucleotide repeat mutations. And there's insertion of a novel transcriptional promoter, for example in viral cases, which could also lead to, there's also another mechanism of activation too. So in summary, when it comes to oncogene activation, electrochemical, carcinogens, radiation, infectious agent, leads to DNA damage. There's a transformation, whether through point mutation, translocation, or amplification, and or if there's an insertion of a new growth promoter. Right, so normal cells, growth factor, signal transaction, growth factor receptor, there's activation of transcription. Neoplastic cells are being formed. Increasing the growth factors will of course there'll be increase in growth factor receptors, which will increase the signal transduction and increase in activation of transcription. So when it comes to viral oncogenes, the viral oncogenes, or the VONCs, they originate from cellular proto-oncogenes, and the current view is that the viral oncogenes arise as a result of transcription from a cellular proto-oncogene that's that particular cellular genetic material has been incorporated into the viral genome, probably through an integrase. So with tumor suppressor genes, they encode negative transcriptional regulators, and they are like negative transcriptional regulators of the cell cycle, and both alleles must be inactivated to produce the deficit that allows tumor development. So heterozygote state is sufficient to protect against cancer. So lots of heterozygosity predisposes to tumor development. So with tumor suppressor genes, almost all tumors have defects, and in the tumor suppressor genes, and certain hereditary tumors are associated with mutations or loss of alleles in germline cells, like retinoblastoma, of course, RB gene, the 13q14, and Worm's tumor, Worm's tumor 1 gene, that's 11p13, and neurofibromatosis type 1, that's the NF1 or 17q112, and familial adenomatous polyposis coli, that one is in 5q1522. Alright, for tumor suppressor gene like p53, The mutation appears to be the commonest abnormality related to tumor suppressor genes in human neoplasms, and it causes arrest in the gene 1 phase in cells, of course, in the cell cycle with damaged DNA to allow time for excision and repair. But the p53 protein can induce apoptosis, which eliminates the cell's aberrant genomic abnormalities. But if there's a problem with this, you know what can happen. So if there's a DNA damage, there's an increased p53, and then the cell cycle is arrested in gene 1 phase, and there's DNA repair, or induction of apoptosis and elimination of the cells from which the mutant clones arise. Yeah, so then let's take human papillomavirus comes in. The viral proteins have effect on these tumor suppressors. So human papillomavirus release can cause an inactivation of these tumor suppressor genes. So let's say the protein products from HPV, human papillomavirus, or the viral proteins like E7 will inactivate retinoblastoma tumor suppressor gene, and E6 will inactivate the tumor suppressor gene P53. The serotypes or the types of human papillomavirus that causes this is cysteine, HPV 16 and 18, leading to carcinoma. Yeah, so properties of oncogenes and tumor suppressor genes. When there's an activated oncogene, it acts dominantly. Inactivated tumor suppressor genes and also activated oncogene will have two pathways. So let's take the inactivated or loss of function of the tumor suppressor genes. It acts, let's say it's recessive, and it produces, there's a loss of function, and that could lead to neoplasia. When there's an activated oncogene like proto-oncogene leading to gain of function of the proto-oncogenes, and when it acts dominantly, even if it's one lesion, produce gain of function, lead to neoplasia. Okay, so genes that regulate apoptosis, there are a large family of proteins inhibitors of apoptosis. So it's the BCL2, BCL extra large XL, and the promoters of apoptosis, BAX and BAD, and BCLXS. Yeah, so inhibitors are BCL2 and BCLXL, and then the promoters are BAX and BAD, BAX, BAD, and BCLSXS. Yeah, so other cancer-associated genes intimately connected with apoptosis are the p53 gene I talked about, and the proto-oncogene like CMYC, and activation of p53 upregulates BAX. BAX are promoters of apoptosis, so BAX synthesis, and also counteracts the anti-apoptotic action of BCL2. Remember BCL2 and BCLXL. Yeah, so CMYC induces apoptosis when growth driven by CMYC is restricted by limited availability of growth factors in the tissue, and p53 is then upregulated. Okay, so the DNA repair genes are responsible for repairing any damage to the genetic material and extremely important in maintaining the integrity of the genome. Defects in these genes results in an increased susceptibility to tumor formation, and defects can be inherited or sporadic. So for DNA repair genes, there are several types of DNA repair genes, whether DNA mismatch repair genes, nucleotide excision repair genes. For DNA mismatch repair genes, it acts as proofreaders during replication, for example in HNPCC syndrome. And for nucleotide excision repair genes, it repairs pyrimidine residues that are cross-linked in UV light, for example in xeroderma pigmentosum. And for recombinant repair genes, they repair DNA damage due to many agents, for example, ionizer radiation, DNA cross-linking agents. So a group of autosomal recessive disorders are characterized by defects in these genes, for example, Bloom syndrome, Fanconi anemia, atalaxia, telangiectasia. So let's look at um, telomerase and cancer. So the scene follows a fixed number of division of normal cells, and each cell division leads to shortening of specialized structures at end of chromosomes, like um, uh, that's the telomeres. So beyond a certain length, there is lots of telomere function, and end-to-end fusion of chromosomes cell leads to cell death. So germ cell shortening is prevented by function of telomerase. That's the enzyme, and enzyme is absent from most somatic cells. Tumor cells reactivate telomerase activity in affected cells, and other telomerase lengthening mechanisms have been found in some tumor cells. So let's take viruses, chemicals, radiation, physical agents, inherited mutations in DNA repair enzymes leads to mutations. There's an activation of tumor suppressor genes, activation of the oncogenes, increased sensitivity to um, apoptosis. There's aberration, aberrant DNA and histone modification or methylation, and malignant transformation, clonal expansion, tumor heterogeneity, invasion, and finally metastasis. Yeah, so this concludes this section. We'll continue with the next tutorial soon.